Hello, my name is Piyush Kumar Pathak, and in this video I am going to discuss one of the very important topic from zoology, and that is enzymes. This many questions are being asked from this topic, and this topic is very conceptual point uh, from understanding point of view, and so many questions are being asked in NEET examination. So let's dive deep into the topic and understand the basics of this topic. Okay. So we have heard about the word enzymes in our day-to-day life, and we will also see a lot many enzymes in this biology. From the very beginning of the biology, we will see many enzymes in the digestive system, in the nervous system, in the endocrine system, and in the excretory system. So there are a lot many enzymes mentioned in this chapter, and we have also heard this word in our day-to-day life. So what are enzymes? So enzymes are basically catalyst. Enzymes are basically catalyst, and they are catalyzing reactions inside a cell, inside a living system, inside an organism. So they are a biocatalyst. So enzymes are biocatalysts that are catalyzing, they are speeding up reactions inside living systems. So they are called biocatalysts. Okay. We also have inorganic catalysts that are also catalyzing the reactions in as we have seen in the chemistry. So what are the differences between an enzyme and an inorganic catalyst? The most and foremost difference is that temperature. The enzymes are unable to perform at a very high temperature, very elevated temperature, but inorganic catalyst can perform at a very high temperature. And the second point is that the enzymes are very substrate and reaction specific, but inorganic catalysts are not that specific. For example, iron. Iron is an enzyme. Sorry, iron is an inorganic catalyst and it catalyzes different many reactions. But that is not the case of an enzyme for like pepsin. So pepsin is an enzyme that breaks protein in a stomach, and it will always break protein and it won't take part in any other reaction. So we see that enzymes are very substrate and reaction specific, but inorganic catalyst can catalyze variety of reactions. Okay, so so we now see that enzymes are very important for the functioning of life. All the reaction, there are millions of reactions taking place inside the body, inside the cell, inside an organism, and they are very important to maintain the life. So we can say that enzymes are central to the functioning of cell, and of course, obviously, it is important for the existence of life. Without enzymes, life is not possible because each and every reaction is catalyzed by an enzyme. If we talk about the nature of enzymes, so enzymes are generally proteinaceous in nature. All enzymes are not proteinaceous in nature. Recently, some enzymes have been discovered that are not proteinaceous in nature, and they are they are called ribozymes. So they are ribozymes because they are made up of RNA. So here RNA is acting as an enzyme. One example is RNAsP, ribonuclease P that helps in breaking the RNA. Another example we have RNA synthase enzyme because these enzymes form rRNA from proteins. So they are also not proteinaceous in nature. Okay, so we have talked about the nature of enzymes. So enzymes are proteinaceous in nature, and since they are proteinaceous in nature, they will have their primary structure, they will have their secondary structure, and they will have their tertiary structure because all the proteins have their primary, secondary, tertiary structure. But if we talk about the functionality of enzymes, so enzymes are functional in their tertiary structure. So because tertiary, in tertiary structure, the chain of polypeptide will fold upon itself, and during this folding, many crevices and many structures will be formed on the surface, and those surfaces are responsible for the functioning of enzyme. We will talk about these structures later on. So we now see that enzymes are proteinaceous in nature. All enzymes are not proteinaceous in nature. Some are made up of different chemicals and different chemical characteristics, and such as ribozymes. So this is very important. And enzymes are functional in their tertiary structure. And the last point is that I have already discussed, enzymes are highly specific and highly efficient. To understand this, we can take an example of carbonic anhydrase. Carbonic anhydrase. So carbonic anhydrase is an enzyme that catalyzes this reaction, CO2 plus H2O, and inside the RBC it forms hydrogen bicarbonate. So normally the speed of this reaction is 200 molecules per hour. So if this reaction is uncatalyzed, then the speed of this reaction would be 200 molecules per hour. But when the same reaction is catalyzed, the speed of the reaction becomes 3600 molecules per second. So we see that the speed of the reaction has almost becomes 10 million times. 10 million times. So we see that enzymes are very efficient and very fast. Sorry, it's 3600 molecules. Anyways, so we see that enzymes are very fast and very efficient as compared to the inorganic catalyst. So uncatalyzed reaction and catalyzed reaction show very far differences as compared to the uh, normal reactions. Okay, we see that the enzymes are catalyzing reactions, but we need to understand how this happens in terms of mechanism. So if we try to understand the mechanism of a reaction, this graph will discuss as the mechanism of reaction. So this is a graph that shows the potential energy. The y-axis is the potential energy of the substrate and product, and the y-axis, x-axis is the progression of reaction. So we see that this is the potential energy of the substrate, and from here the energy of the substrate is increasing to a certain threshold, and finally this it decreases to form the product. So what's happening, we need to understand here. So suppose this is our enzyme and this is our substrate. So what happens, first the substrate diffuses to the crevices that we talked about, the structures that are formed due to folding of enzymes, folding of polypeptide structure. So the structure is called active site. So this is our enzyme and this is our substrate. So substrate diffuses to the active site of the enzyme, and there is a formation of enzyme substrate complex. Here there is a bond breaking and bond making process, and during this process many transition states are formed. But many transition states are formed that are temporary and unstable. But this formation, why does this transformation takes place? Because during the conversion of substrate to product, many transition states are formed during the bond making and bond breaking, and finally the substrate is converted to product. So this transition state requires energy. We know that any reaction is to start a reaction we need to apply some energy. So this energy will increase the energy of substrate. This energy that we supply will increase the energy of the substrate to a certain threshold from the initial energy, and after that once the transition state is formed that is very unstable, they will degrade and break down into products. So this rise in energy from the initial state is called activation energy. We have seen about the activation energy. We will read about activation energy in chemistry. So let's understand this activation energy. So activation energy is basically the energy that is supplied to the reaction. We will learn about this in Arrhenius equation. So the energy supplied from the environment or from the outside basically increases the collision probability, and that fruitful collision that will lead to the formation of product will increase. So if we increase the energy, what will happen? The number of fruitful that in better conformation, in better configuration will increase, the number of successful collision will increase, and that will lead to the formation of product. So high energy is basically signifying that number of successful collisions. Okay, so this is the activation energy. Now here comes the role of enzymes. Enzymes, what it does, it simply decreases the need of this energy, activation energy. So this, the requirement of this activation energy is decreased to very low quantity that can be supplied within this active site, and the product can be found very easily. So so we see in the graph that this is the activation energy without enzyme, and this is the activation energy with enzyme. So with the help of enzyme, the requirement of energy is decreased and the product can be found very easily and very quickly without supplying much energy. Another thing that we can conclude from this graph is that will the reaction be exergonic or endergonic? So if the difference between the substrate energy level and product potential energy will be positive, that means if substrate has high energy compared to the product, then the extra energy will be released and the reaction will be exothermic. Exothermic. And if the energy of the product, for suppose example, we have like this, we have a graph like this. So if the energy of the product is more than the substrate, then extra energy have to be supplied and the nature of reaction will be endothermic. So two things we can understand from this graph, exothermic and endothermic, and what is the role of enzyme. So enzymes function by decreasing the requirement of activation energy, and activation energy is basically the energy required to increase the number of successful collision in product formation, that we will see in class 12th chemistry, that is Arrhenius equation. So in there the concept of activation energy is explained in detail. So that was a part of chemistry, but we are in the biology. So we will understand simply what activation energy is, the energy difference between the substrate energy and the energy of transition state. Transition state is temporary and unstable. That's why it breaks into product. Okay. And in the last, the products released will be released with absorption or release of energy that I have already discussed. So this was all about the mechanism of enzyme action, and hope we have understood the points. Now let's move on to the next point, that okay, I understand how the enzyme is speeding up the reaction, but what are the, how does it happens, how the enzymes does this? So to explain these things, two theories were given, put forward, that are most accepted, and first is lock and key hypothesis and second is induced field theory. So lock and key hypothesis was given by Emil Fisher, and it considers that the active site is a rigid structure, it won't change, and this active site will be complemented by, okay, and this active site will be, okay, so this is our enzyme and this is our substrate. So this active site is complemented by the product, sorry, substrate will fit into this active site and only a certain substrate can fit, just like lock and key. Suppose this is the enzyme is the lock and this is the key. So only one key can open a lock, and a particular key can open a lock. Similarly, only a particular substrate can fit into the active site and it can get converted into the product. So this was a hypothesis given by Fisher. So this is called lock and key. So we know the structure of the crevices of the lock won't change. So it consider the active site as a rigid structure. But there were certain drawbacks. There were some questions that this theory cannot answer. So what was that? So if only fitting of the size is the point, then anything like this substrate will fit into the active site and will get converted in the product. But this won't happen. This does not happen. Anything other than that, other than the substrate gets into the way, it won't proceed forward and no product will be formed. So lock and key hypothesis, why is this happening, won't answer. So to answer this question, that why anything that is similar to analogous to substrate is not proceeding forward into product. To explain these points, another theory was put forward and it is called induced fit theory. It was given by Coastland and it considers that active site is not rigid, rather it is flexible, and approach of substrate, arrival of substrate induces the active site to fit firmly around it. Okay, so see in the diagram that this is the substrate, and we see that the active site is not according to the structure of this substrate, but when the substrate arrives, this active sites, it induces the active site to change its conformation and fit properly, fit around the active site, fit around the substrate, and finally the substrate is converted into the product. So what happens that this substrate has certain groups over the certain molecules, certain atoms above the surface that induces the uh, amino acids around the active site to change their conformation. So that these are called buttressing groups. So buttressing groups help to change the conformation of the enzyme to uh, bring about the conformational change and convert the substrate into the product. So if we supply another product, another substrate that is similar to this substrate, it won't have those molecules on its surface and it won't be able to induce the active site to change its conformation, so the reaction is not proceeded forward. It is not fit firmly in the active site. So it explains the drawback of the induced lock and key hypothesis. Okay, so this was the theory given by Coastland, induced fit theory. So now we understand that how the enzymes work, how it speeds up a reaction. So we will understand now the factors affecting the enzyme. So the first and foremost factor is the temperature. So there is a general thumb rule that if we change the temperature by 10 degrees centigrade, it will either double the reaction if we increase it, or if we decrease by 10 degree it will become half. So if we double the reaction it will become double, and if we increase by 10 degrees centigrade it will become double, and if we decrease by 10 degrees centigrade it will become half. But there is an upper limit. We after a limit, so for 37 degree in our case, so if we increase the temperature over 37 degrees centigrade and we expect that okay the reaction will become double, this won't happen, because after a certain upper limit, certain temperature, that is optimum temperature, if we increase the temperature it will leads to denaturation of the enzyme, and the enzyme activity will be decreased. Similarly, if we decrease the temperature to a very low temperature, the functioning of be seized and the enzyme will be temporarily inactive. So we can store the enzyme in inactive form for long duration under very low temperature, say for minus 20 degrees centigrade, minus 80 degrees centigrade, we can store the enzyme. So this is a very important point, that at very high temperature why the functioning of enzyme is uh, decreases due to denaturation of the enzyme, because enzyme is a protein, we have discussed it earlier. So the proteins will denature, their tertiary structure want to be stable at a very high temperature. But in case of certain bacteria and certain organisms that live at very high temperature, their enzymes are functional at very high temperatures. For example, that Thermophilus aquaticus. Thermophilus aquaticus. From those we have obtained Taq polymerase. So Taq polymerase is a polymerase enzyme that is functional at very high temperature, because this bacteria is found in hot springs. So they have enzymes that are functional at, as also we have some enzymes that are functional at very low temperature also, that are called chelating proteins. Chelating proteins, they can function at very low temperatures. For example, bacteria living in polar ice caps. Okay. So this was about the first factor, that is temperature. The second factor is pH. The enzyme is very sensitive to pH. They are functional at a very particular pH, that is optimum pH, and before, above or below that optimum pH they are non-functional. For example, if we take the example of pepsin. Pepsin is an enzyme that is functional at a low pH of a stomach, say 2 pH of 2. So if the food that is present in the stomach moves to the intestine where the pH is say 8, so the pepsin present in the food won't be functional at this pH. So the pepsin enzyme is no longer active in the intestine. So this happens due to the change in pH. So enzymes are very sensitive to pH. As pH change will render the enzyme non-functional, and they are functional at a particular pH, that is called optimum pH. So in the graph we can see uh, before and after the optimum pH, the uh, enzyme activity is decreased. Okay. The third is the substrate. This is very important. So the first way, general thumb rule is that if we increase the substrate concentration initially, the rate of reaction will increase, and after a certain time it will become constant. So to answer this, why this is becoming constant, two scientists, Michaelis and Menten, they worked on the enzyme kinetics, and they presented their work in the form of an equation that is called Michaelis-Menten equation. So the Michaelis-Menten equation basically deals with the effect of substrate concentration on the rate of reaction. So what happens? So So the answer to this question is that by it becoming constant, that initially we have free enzymes. So we have enzymes, and we have substrate. So initially what happened? The enzymes are free, the substrate are getting bound to the enzyme. So initially the rate of reaction increases because enzymes are free. After a certain time, all the enzymes become engaged. They are now, their active site are occupied by the substrate. So any further increase in the substrate concentration will have no change on the, because no free enzyme is available. Once the enzyme is free, they will bind to the substrate. So initially the rate of reaction increases and then it becomes constant, and we have this rectangular hyperbola graph. Okay, so this is the Michaelis-Menten equation. So in this Michaelis-Menten equation, this is V, velocity. So if the reaction is taking place in a particular direction, we can consider it as a velocity of reaction or rate of. So rate of reaction can be considered as a velocity of reaction. So this is the V, velocity of reaction, and this is the V max, maximum velocity that is achieved by an enzyme. This is the S is the substrate concentration, Km is very important. This is called Michaelis-Menten constant. So this is the constant of the equation, and basically Km is the substrate concentration. Km is the substrate concentration at which maximum half of the maximum velocity is obtained. So Km is Michaelis-Menten constant. So it is a substrate concentration at which half of the maximum velocity is obtained. For example, in this graph, we have this is the maximum velocity, and the velocity at which the half of the maximum velocity, the substrate concentration at which half of the maximum velocity obtained is the Km. So Km is what? Substrate concentration. So the units of substrate, unit of Km will be the unit of concentration. Okay, I hope this is clear. So this was the effect of substrate on the enzyme action. Okay, now let's move into a very important topic, that is enzyme inhibition. So we have seen that there is a substrate, there is an enzyme, there is a formation of enzyme substrate complex, and finally the substrate is converted into the transition state, then products, and the products are released. This is the general thumb rule. So what is the general thumb rule? What happens? We have enzyme and we have substrate, then there is a formation of enzyme substrate complex, and then we have enzyme product complex, and then enzyme products are released and enzymes can become free and it can be utilized. But there is, it is not always that substrate, there is only substrate around the enzyme. There are other molecules around the enzymes that can also bind to the enzyme, and they can either affect the enzyme or affect the reaction. So those are called inhibitors. So what will happen? If any other molecule other than substrates binds to the enzyme or uh, any structure in the process, and it decreases the performance of enzyme, then it is called enzyme inhibition. So this is the types of inhibition. So we have, inhibitors or inhibition can be categorized into broadly three categories, that is reversible, irreversible, and allosteric. So allosteric is also called feedback feedback inhibition. Okay, so uh, we are much concerned about this reversible reaction because they are being asked in NEET examination, but this allosteric and reversible are rarely asked. But I will explain few about also them also. So there are three types of reversible. One is competitive, second is non-competitive, and third is uncompetitive. So first is competitive. So what happens in competitive inhibition? Okay. So this is competitive inhibition. So in case of competitive inhibition, what happens? There is another molecule that is called inhibitor, and this inhibitor is similar to that substrate, and it can also bind to the active site of the substrate. So there is a competition for active site between the substrate and the inhibitor for the active site of the enzyme, and if this inhibitor binds to this active site, the substrate won't be able to bind to the active site of the enzyme, and so no product will be obtained. So this kind of inhibition is called competitive inhibition. So we see that this inhibitor is binding to the free enzyme, and it is forming enzyme inhibitor complex, and substrate is not able to bind to the enzyme. So in the very initial stage, the inhibitor binds. So inhibitor is also how we can overcome? So this is a reversible reaction. So we can overcome this inhibition. So how we can overcome? If we supply more substrate, then the probability of substrate binding to the active site will increase, and thus we will get our product. So this type of inhibition can be overcome by increasing substrate concentration. Okay, so if we talk about the example of competitive inhibition, then we have very two famous competitive examples of competitive inhibition. One is inhibition of succinate dehydrogenase. Succinate dehydrogenase is an enzyme that is found on the inner mitochondrial membrane. So we will learn about this in uh, uh, respiration chapter. So this enzyme is inhibited by malonate. Malonate. So malonate is an analog to the normal succinic acid, and it binds to the succinate dehydrogenase, and it inhibits the succinate dehydrogenase. Another is very common example, that is ethanol inhibiting alcoholic dehydro dehydrogenase. So in what happens in case of methanol poisoning, there in methanol poisoning, like some people you accidentally take that poisonous alcohol, and that causes the blindness and other things. So we can overcome that effect of poisoning, methanol poisoning, by adding ethanol. So ethanol will compete with the methanol for the active site of alcoholic dehydrogenase, and it will cure the poisoning effect of that methanol. So this is also an example of competitive inhibition. So these two examples are very common and they are asked in the examination. Another thing that you need to consider is that what happens to the Km value and Vmax. So in case of competitive inhibition, we see that to overcome the inhibition, we need to supply more substrate. So what will happen? Km value will Km value will increase, but Vmax will remain constant, because nothing is happening to the enzyme. So Km value will remain constant, but Km value will increase, but Vmax will be constant. There is no change in the Vmax. Okay. Next inhibition is non-competitive inhibition. In this inhibition, the inhibitor is not competing for the active site. Rather, it binds to another site present on the enzyme, and it changes the conformation of the active site so that substrate cannot bind. So what is happening? Listen again. The inhibitor is binding to another site on the surface of enzyme, and it brings about conformational changes of the active site so that substrate cannot bind. So this is making the enzyme unreceptible for the substrate. So the conformation of the active site is changed and substrate cannot bind to it. So how can we overcome? So we cannot overcome this inhibition by increasing the substrate concentration, because it is binding to the enzyme. There is no relation with the substrate. Okay. So example of non-competitive inhibition is organosuccinate, organophosphate. So it is in the case of nerve gas. So nerve gas. So nerve gas, it was used in the World War. So nerve gas, what it contains? It contains organophosphate. So these organophosphate bind to the enzyme acetylcholine esterase. What is the name of the enzyme? Acetylcholine esterase. So this enzyme is responsible for breaking of acetylcholine, the neurotransmitter, and breaks the signal after the reception in the postsynaptic membrane. But this organophosphate binds to the acetylcholine esterase enzyme, and inhibits it. So the acetylcholine is not uh, degraded, and what happens? There is a continuous impulse formed in the nervous system. This leads to death. So this gas, organic phosphate, Organophosphatase is a class of chemicals. They are found in the nerve gas, was used in the World War. Okay. So let's see here the what is the value of Km. So the value of Km will not change. So value of Km will be constant, but Vmax will decrease. Okay. So we need to understand this point. And in the third is no uncompetitive inhibition. In uncompetitive inhibition, what is happening? The enzyme is binding to the enzyme substrate complex. Okay, the enzyme is binding to the enzyme substrate complex. So it is taking out the activated enzyme outside the reaction. So there is also no relation with the. So now the substrate is bound to the enzyme. So and but it is not getting converted into the product. Okay, so this is the uncompetitive inhibition. Example of uncompetitive inhibition is that so example of uncompetitive inhibition is okay. So we have example of uncompetitive inhibition as fluoride poisoning. So fluorides basically uh, inhibits the enzyme hexokinase. So it inhibits the enzyme hexokinase. It binds to the enzyme substrate complex. Okay. Now let's look at the Km and Vmax value. What is happening? Km is also decreased because now less substrate is required because enzymes are less, so less substrate is required to reach the half of the maximum, and also because maximum velocity is decreased. So both Km and Vmax are decreased. Okay. So this was all about enzyme inhibition. This is the suicidal inhibitor. What happens in case of suicide inhibitor? The enzyme binds permanently to the enzyme and it makes the enzyme dysfunctional, non-functional. The example of suicide inhibition is cyanide poisoning. So cyanide poisoning is an example of the cyanide poisoning, an example of suicidal inhibition. And third, last is allosteric inhibition or feedback inhibition. So what happens in feedback inhibition is that so in case of feedback inhibition, suppose this is A, this is acted by an enzyme E and it forms the product P. So this product P will bind to the enzyme and it will inhibit the enzyme from further processing this reaction. So if the amount of product is very much, it becomes very potent in nature and it inhibits the enzyme. So this is also called feedback inhibition. So the enzyme is giving feedback, so this product is giving feedback to the enzyme that okay, the amount of product is enough and we not need to produce much enzyme. Next example is the example hexokinase. So the product of this enzyme, that is glucose 6 phosphate, it inhibits the hexokinase to further produce the glucose 6 phosphate in glycolysis. Okay, the very first reaction of glycolysis. So this was all about enzyme inhibition. I hope it is clear. Okay, so now this is the another important topic. So there are so many enzymes and it is very difficult to remember them. So enzyme commission, there is a commission that is called enzyme commission. Enzyme commission. So enzyme commission in our organization that what they have done, they have categorized all the enzymes into 6 classes. There are 6 classes of enzymes and this order is cannot be changed. This is fixed order. So and they have named the enzymes with certain numbers, that is called enzyme commission number, EC number. So each enzyme has their own EC number that is unique to them, and it tells that what is the function of enzyme, which class it belongs. So every details of that enzyme is represented by that enzyme commission EC number. So this group 1, 2, 3, these numbers, these are basically the first number of that nomenclature system. So this number cannot, the sequence cannot be changed. This is fixed. So the first class is oxidoreductase. So what happens in case of oxidoreductase? Oxidoreductase class of enzyme, what they does? They carry out oxidation reduction reactions. So transfer of electron or H plus. Transfer of H plus is also an oxidation reduction reaction. So we see here in the example there is an molecule that has a minus charge and this is neutral. So this minus charge has been transferred to the B. So basically one H plus has been transferred from the B to A, or we can say one electron has been transferred from A to B. So this is an example of oxidation reduction. So one molecule is oxidized, another is reduced. Best example is alcoholic dehydrogenase. What is that? It oxidizes alcohol to acid. So this is an oxidation reaction, acid alcohol to acid. Next is transferase. They transfer group, but the point is that it should not contain H plus, because if they transfer a H plus, this it will be a oxidation reaction, redox reaction. So transfer a group except H plus. So they can transfer carbon, nitrogen, phosphate. For example, we have this AB and now this B is transferred to C. Example is hexokinase. What is this? It transfer a phosphate group to glucose and it forms glucose 6 phosphate in the first step of glycolysis. Okay. Next is hydrolases. What they does? They cleave the bond in the presence of water or addition of water. So they cleave a molecule. They cleave a bond with the help of water. So here AB is a molecule and with the help of water they are basically hydrolyzing the molecule. Example is esterase. What is that? Esterase, it breaks ester into alcohol and acid. Okay, we have trypsin. What is that? It breaks proteases, proteins with the help of water into amino acids. So hydrolysis of proteases and peptones with the help of water into amino acids. So this is done by trypsin. So this is a kind of hydrolysis reaction. Next is lyases. They are also breaking the bond, but here water is not present. So water is absent. So if we use water, then it will be hydrolysis. So without the presence of water. So they are also breaking the bonds like CC bond, CS bond and CN bonds to form a double bond. For example, we have this AB, so C and D are removed and now become double bond. Example, pyruvate decarboxylase. What is that? It removes the CO2 molecule from pyruvic acid and forms acetyl coenzyme A. So it is removing a CO2 molecule from pyruvic acid. Okay, so this becomes acetyl coenzyme A. Okay, so this was lies. Third is isomerase. They form isomers, either optical isomer or geometrical isomers. Example, we have C and D. Now this D is coming here attached to A, and so there is the molecule is not changed, but the conformation has changed. So this is an isomers. Example, we have many isomerase enzyme in the glycolysis, also in the Calvin cycle also. So there are many isomerases. One is also fructose isomerase, that DHAP is converted into phosphoglyceroldehyde. So there is also an isomerase enzyme. So we have malate isomerase. And the sixth is ligases. So what they are doing? They are making bonds. So two molecules are joined by the use of energy. So if we are forming a bond, we will require energy, and that energy is supplied in the form of ATP. ATP is basically our energy currency. So every energy expressed in our body in a living system is met by use of ATP. So use of ATP. The most famous example we have that T4 DNA ligase. T4 DNA ligase. So this ligase enzyme is joining two strands of DNA. Another we have pyruvate carboxylase. So this is adding a CO2 to the pyruvic acid, and it will happen in the case of we will read about this enzyme in photosynthesis. So we see that enzymes are coming from all the chapters of the biology. You see different enzymes from the plant physiology, animal physiology, and different chapters we are getting the enzymes. So we will go look into these enzymes when we will read those chapters individually. And the last topic is that types of enzyme on the basis of chemical composition. So till now we have seen that enzymes are formed of only proteins, but this is not always the case. So proteins sometimes contains other things also, and those proteins that contain any other molecule or groups that are not proteinaceous in nature, then those proteins are called conjugated proteins. So in case of conjugated proteins, the protein part, this is the protein part. So the protein part is called apoenzyme and the non-protein part is called cofactor. Non-protein part is called cofactor. So, and when these two are joined to form the functional enzyme, this is called holoenzyme. This is the functional enzyme. So this is the functional enzyme. Okay, so we will look at the cofactors. So holoenzyme equal to apoenzyme plus cofactors. Without the cofactors, this holoenzyme is not formed and it is not functional. So we have two types of cofactors. One are ions, other are coenzymes. So ions we have metal ions and coenzymes are other organic molecules. So if the ion is loosely bound to the enzyme, then it is activator ion, and if it is tightly bound, then it will form metalloenzymes. So we have some enzymes that requires uh, permanent attachment of a metal ion for its functionality. And another we have coenzymes. So if it is loosely bound, it is organic molecule. Organic molecules. So if we have loosely bound, then they are called co-substrates, and if they are tightly bound, they are called prosthetic groups. That's it. So it was all about enzymes, and now we will look at some questions asked from this topic in the NEET examination. And okay, so the first question: consider the following statement. Coenzymes or metal ions that is tightly bound to the enzyme protein is called prosthetic group. Just now we have seen it. Tightly bound prosthetic group. First statement. Second, a complete catalytic active enzyme with its bound prosthetic group is called apoenzyme. So apoenzyme is not the complete enzyme, it is the protein part. Without the cofactor, they are not functional. So a complete, this is not complete. Apoenzyme is not complete. So this statement is wrong. And prosthetic group, okay, the prosthetic group are tightly bound, so this statement is correct. So A is correct, B is false, so second option is right. It is wrong, you have to erase it. Okay, okay, now let's look at the second question. Which of the following statement regarding enzyme inhibition is correct? We are talking about enzyme inhibition. Competitive inhibition is seen when a substrate competes with an enzyme for the binding to an inhibitor. Substrate is not competing with the enzyme, it is competing with the inhibitor. So this statement is wrong. Second, competitive inhibition is seen when the substrate and the inhibitor compete for the active site of the enzyme. Competitive inhibition is seen when the substrate and the inhibitor compete for the active site. Okay, this one is right. We'll keep it for further. Non-competitive inhibition of enzyme can be overcome by adding large amount of substrate. Non-competitive inhibition. So in case of non-competitive inhibition, it cannot be overcome by adding more substrate. So this statement is wrong. So this one is wrong. And so which one is correct? Only second. Second option is correct. Okay. Now the third question. Transition state structure of the substrate formed during an enzyme reaction is. Okay, I have talked about the transition state. They are formed temporarily and they are unstable. So first option is transient but stable. This is not stable. Permanent but unstable. This is not permanent. So this one is also wrong. Transient but unstable. This is correct. Permanent. This is not. So the right answer is 3. Third option. Okay. Now let's move to next question. Select the option which is not correct with respect to enzyme action. Okay. Substrate bind with enzyme at its active site. Okay, this one is right. Addition of a lot of succinate does not reverse the inhibition of succinate dehydrogenase by malonate. So we have talked about succinate dehydrogenase. So this is an example of competitive inhibition. So in case of competitive inhibition, if we add more substrate, then we can overcome the inhibition. So this statement says that it does not overcome. So this statement is wrong. So we have to correct the wrong statement. We have to choose the wrong statement. Okay, third option. A non-competitive inhibitor binds the enzyme at a site distinct from that which binds the substrate. Okay, a non-competitive inhibitor binds the substrate at a site distinct from that which binds the substrate. So in case of non-competitive inhibitor, what we have seen, that the inhibitor is binding at a site other than the active site. So this statement seems correct. At a site distinct from that which binds the substrate. So this statement is correct. Okay. Malonate is a competitive inhibitor of succinate dehydrogenase. So this is right. This is right. Malonate is a competitive inhibitor of succinate. This is also right. So this one is the wrong statement and this is our right answer. This statement is wrong, but the answer is right because we have to choose the wrong statement. Okay. Km value is dependent upon. We have seen that Km value is dependent upon. What is Km value? It is the substrate concentration at which half of the maximum velocity is obtained. So this is a substrate concentration and it is dependent on substrate concentration. So it has nothing to do with temperature and nothing to do with the enzyme concentration. Because for a particular reaction, for a particular enzyme, it is constant. That's why it's Km constant. Michaelis Menten constant. Okay, next question we have non-competitive. In non-competitive inhibition, the substrate inhibitor cannot bind to the enzyme at the same time. Then what happens in case of non-competitive? It binds to a site other than the active site. So the substrate inhibitor cannot bind to the enzyme at the same time. It can bind because they are binding to the another site. So this is wrong. The inhibitor binds only to the substrate enzyme complex. That is the case of non-uncompetitive inhibition. That substrate complex. The inhibitor can bind to the enzyme at the same time as the enzyme in substrate. This one is right. And the fourth, the binding of the inhibitor to the enzyme reduces its activity but does not affect the binding of substrate. This affects the binding of substrate. We have seen that it brings about conformational changes to the active site and substrate cannot bind. So this one is also wrong. So we have the right answer is third. Okay. When the cofactor is removed from the enzyme, the catalytic activity. So we have seen about the cofactors. If they are not attached to the conjugate enzyme, that is apoenzyme, then the functionality of the enzyme is lost. So it is not reduced, not increased, not unaffected. It's lost, complete lost. Okay. So these were some questions that were from previous examinations of NEET, previous years of NEET, and I hope this helps to understand the topic better. And that's all from this topic. So thank you.